Welcome back, everyone. Great to see the room filling up, and hopefully you're all stretched out and you've let your body have some nutrition. This is my one link that I've managed to pull together here, because we're going to, the human body's not really designed for other planets. We're really designed for Earth's operating conditions with gravity, water, sunlight, and so forth. And so what happens when we head out into space? Well, we have been testing what happens in other settings on Earth, such as Antarctica, where it's dark for most of the year. We're wondering, what does that do to our vitamin D? How do our eyes behave? So we're really grateful and lucky, and I'm super excited to introduce Dr. Scott Smith. Hi, Scott, who's a nutritionist at the NASA Johnson Space Center. And so I'm going to hand over to Dr. Scott Smith. Please make him welcome. Thank you, Scott.
Thank you. I want to thank the organizers of the meeting. It's an honor to be here, and it's great to talk to you all down there. And good morning in Sydney. Again, what I'd like to try to do today is to give you a bit of an overview of the work that we're doing here at the Johnson Space Center in Houston. And in particular in the lab that I run along with my colleague, Dr. Sarah Zwart, and that is the Nutritional Biochemistry Lab. We have a few goals, but one of our leading goals is to help keep the astronauts healthy during spaceflight from a nutrition point of view. And at this point, the primary focus of our work is the astronauts that are on board the International Space Station right now. We just passed 22 years of continuous human presence off the planet. And we've literally worked with all of those crew members to help guide them in the right direction and to help better understand how their bodies change during flight. And again, but I want to try to give you a bit of an overview of what we do and kind of a bit of a deep dive into a few of the key areas where we found some interesting things. So we do two types of work in the Nutritional Biochemistry Lab. The first we call operations, which is work, it's essentially clinical work that we do for the flight surgeons. And again, we're working to keep the astronauts healthy. We do a nutritional assessment workup before flight, where we collect blood, we collect urine, and we analyze those for a whole host of biochemicals, vitamins, minerals, proteins, bone markers, kidney stone risk, antioxidants, lipids, you name it. We do that twice before flight, and then when the crew gets back, we do it again twice after flight. The goal pre-flight is to make sure that we send crews up as healthy as can be, and post-flight, the goal is to assess any decrements that have occurred and to work with the rehabilitation team to get the crews back to full health as quickly as possible. And to give you an idea where we are right now, tomorrow we will do the testing on some of the Crew 4 astronauts that landed in the SpaceX vehicle. This is their R+30 testing, so it's about 30 days since they've landed. And again, we're geared up to go collect blood from them tomorrow. In flight, we do two things. One is we track body mass, and we do that with very special devices. The one in the picture in the middle there is Karen Nyberg on what we call the SLAM-D. Everything at NASA has to have an acronym. That is the Space Linear Acceleration Mass Measuring Device, which is a scale. And it uses the physics of force equals mass times acceleration to determine a crew member's body mass. And, you know, this is obviously one of the more challenging things to measure somebody's body weight in weightlessness. But when you think about it, when you go to the doctor's office, the first thing they do is put you on a scale. And that's because your overall body mass and changes in your body mass are a very good general indicator of your overall health. So having the ability to do that during spaceflight is critical. The other thing we do during flight is we track dietary intake. We've done this with a few different techniques over the years of International Space Station, but we developed an iPad app that you can see there floating in the cupola on the left. You can see Peggy Whitson had the iPad in her hands there on the right. And that was an app that we used on board Space Station that we called the Food Intake Tracker, or FIT. And we asked the crews to record literally everything that they ate over the course of their typically 6-month missions. And the apps have to be very easy to use. They have to be very user-friendly. They have to provide feedback to the crew. And I think we did pretty well when we built this thing, because in the 6 or so years that we used it on station, we got about 5,000 days of crew use. And we had dietary intake data on more than 90% of those days. So it was a really valuable tool and provided us a lot of data that we're still working on, trying to help better understand the relationships between diet and human adaptation to spaceflight. The other work that we do is research. We are what I call a soft money research lab, which means that we have to scratch and claw for funding like every other research lab in the world. We write proposals, we compete for funding, and every once in a while we get lucky. We do two types of research. We do ground-based research, where we will try to mimic different elements of spaceflight. And depending on what system you want to look at, you use different analogs. So we'll do things like bed rest studies when we want to look at bone loss or muscle loss or cardiovascular changes, where if you put people in bed for a month or 2 or 3, you can start to see deterioration that is analogous to what happens during spaceflight. We've done work looking at oxidative stress in people that are living at the bottom of the ocean for a few weeks. They were in a habitat down there doing some operational work, and what we wanted to look at was changes in things like iron metabolism, vitamin metabolism, and red blood cell metabolism. We've done work in Antarctica for the same reason. If you want to understand vitamin D requirements in people that don't see the sun for 6 months, you go where the sun doesn't come up 6 months out of the year. And we did a couple of studies in Antarctica a few years ago. We didn't study the penguins, but they're cuter than the people we did study. And we studied the crews wintering over at McMurdo Station for 6 months, and learned some very interesting things about vitamin D, not only vitamin D in relation to supplementation, but also vitamin D in relation to its interaction with viral reactivation. We also do chamber studies. This is one of the chambers we have at the Johnson Space Center, where we will lock crews up for, in this case, 30 to 45 days. And they typically use these habitats for behavioral studies, crew cohesion studies. But what the other opportunity is, you have people in a fixed environment. So we recently completed a study looking at different space foods, asking the very basic question of, if we fed people better, would it show up in their physiological outcomes? Those were 45-day studies. We're about to embark on what we're calling the CHAPEA missions. CHAPEA is the Crew Health and Performance Exploration Atmosphere. Sorry. It's a chamber. It's bigger than this one. We're going to put 4 crew members in there for a year each. We're going to do this 3 times. We're always looking for subjects for these studies, so if you're interested, you can find it on the web. We've already picked the crew for the first mission, but there will be 2 others where we'll be looking for votes. And that, to my mind, is one of the most exciting things that we've done in a long time, because that will really get us a look at the long-term implications of eating nothing but space food for, in this case, a year, which will be a major stepping stone to get us to Mars. So our ground research is kind of exciting and unique. Nothing beats flight research. We've had some tremendous opportunities over the years on space station. Nutrition research looks a little bit like the pictures you see here. We track dietary intake. We collect blood samples. You can see in the top left. The second image there is Nicole Stott putting a blood tubes into the centrifuge in the wall to spin them down. The third is a urine collection device. They're not supposed to be free floating, but one of the crew members took it out of the Ziploc, the device that we use to contain it. It took a picture of it floating, because I think he thought it was funny. We don't get many pictures of our urine collection, so we use it. So we collect blood and urine. We put the samples in the freezer that you see Chris Cassidy with on the right there, and the samples will be stored frozen at -96°C until we bring them home. As I mentioned, we started these studies around 2006. We were able to conduct a few studies up until around 2018, and we collected blood and urine samples on about 65 astronauts, which is an incredible number of data points. from spaceflight study. And I want to try to drill into a couple of the key findings that we've had. So one of the things you might have heard about is what we now call SANS, which is a fancy acronym for a syndrome that affects some of the astronauts. That is, some of the astronauts came back from spaceflight with ocular pathologies, eye pathologies, specifically things like optic disc edema or swelling of the disc where the optic nerve enters the eye, cotton wool spots, choroidal folds. And in basic terms, we had some crew members that went up with perfect vision that came back needing glasses. This was first identified about 12 years ago, and it went from we didn't know this was a problem to this is a serious problem and one of our biggest concerns. The thinking when this first happened, and to this day, many people still believe that this is related to environmental factors of spaceflight. The most common one that people believe is that when you go to spaceflight, you're in weightlessness, the fluid shifts from your lower extremities up into your torso and up into your head, and that increased fluid creates pressure, and that the increased pressure in your head pushes on your optic nerve and pushes on your eye, and that leads to these ocular problems. That would be great, and that'd be easy to understand if everybody developed these problems. And the simple fact is they do not. And when this occurred, we started to look at the biochemical data that we had and to see if there was any way to differentiate the crew members that had these eye issues from crew members that did not. And being a bit of a nerd, I can tell you the day, the date, and the hour, my colleague Sarah Zwart came in and said, there's something going on with one carbon metabolism. And what she meant by that was a biochemical pathway that we call one carbon metabolism. It's a very complex pathway, but it's a very important pathway. It occurs in every cell in your body. And it's called this because this is how the body moves single carbon units from one compound to another. It's how you methylate DNA. It's how you convert amino acids from one form into another, and is simply critical for all functions of life. At the center of this pathway is a chemical called homocysteine. Homocysteine is a cardiovascular risk factor. It's an amino acid metabolite, and it occurs in this pathway, which is very nutrition rich. So this pathway involves vitamins like folate and vitamin B6, vitamin B12, riboflavin, biotin, you name it. Excuse me. And what Sarah meant when she said that there was something going on with one carbon metabolism was the graph you see on the right there. And what she'd done is she broke out the astronauts that had eye issues in yellow and those that did not have eye issues in white. And what you can see is that the astronauts with ocular pathologies had higher circulating concentrations of homocysteine. And more strikingly, excuse me, these first three data points were before flight. We looked at this and ruled out as many things as we could. We read the literature and knew that there were genetic influences on homocysteine, and there are genetic influences on this pathway. And what we sought to do a series of studies that I don't have time to show you today, but the bottom line was we proved that genetics and B vitamin status predispose some individuals to develop these pathologies. The key question that remains is whether or not we can use B vitamin supplementation as a way to override individuals' genetics to mitigate this problem. And we've said for almost 12 years now that this needs to be tested, and we are on the verge of doing that. We've got an experiment that we call B complex. You can see the logo there because everything in NASA has to have a logo. And if all stays on track, our first participating astronaut will launch next August, where we're going to give vitamins to counteract this ocular problem. Shifting gears briefly to one of the other interesting areas that we found some exciting data in, is what's called venous thromboembolism, or VTE for short, which in simple words means a blood clot in the veins. The image on the right is what's called Virchow's triad. Virchow was an 18th century physician who postulated that there are multiple factors which cause people to develop blood clots. And those three are shown here: changes in hemodynamics, changes in the coagulability of the blood, and damage to the blood vessels of the endothelium. Well, why is Nutritionist talking about blood clots forming? Well, a couple of years ago, you may have heard, we had an incident on board space station where we had a crew member develop a left internal jugular venous thrombosis, which is a blood clot in one of the veins coming from the head. And that was about as serious as it gets. Again, when we started looking at our data to try to see if there's anything in there that we could understand, we started looking at a protein called albumin. Albumin, I would say, is the vanilla of proteins. It's nobody thinks too much about it. Everybody knows it's there. Half of the protein in your blood is albumin. It has a lot of functions. It's involved in fluid balance, transport of a number of chemicals. It's anti-inflammatory. It protects the blood vessels. And when you don't have enough albumin, or when you have what's called hypoalbuminemia, you can end up with increased blood viscosity, and you can end up with an increased risk of blood clot formation. So what causes hypoalbuminemia? Well, malnutrition is the most obvious thing. That if you're not eating enough, if you're not getting enough protein, you will develop hypoproteinemia and hypoalbuminemia. We knew our crew members were not malnourished. Kidney function changes could be, inflammatory responses could be, but what jumped up to us is that medications can alter albumin synthesis. Specifically, oral contraceptives. And what happens is some of the female astronauts will use oral contraceptives to suppress menstruation during spaceflight, so they don't have to deal with menstruation during spaceflight. We know that oral contraceptives, it's not all of them, that some oral contraceptives have a significant effect on serum albumin. That is, it can lead to hypoalbuminemia. When we looked at our albumin data in that light, what we have is here. On the left, you can see data from the male astronauts, and their albumins did go down a little bit in flight. Not much, but it did go down, but it stayed within normal limits. Women that did not use oral contraceptives, again, their albumin concentrations statistically went down a little bit during flight, but again, it's hard to get too excited about that. But what we noticed here is that women that used oral contraceptives during flight had significantly lower concentrations of albumin than did other crew members, to the point where they were on average outside of normal limits. And of the women we studied in this data group, all of them had at least one data point during flight where their albumin was outside the lower limit for normal albumin. We diagram what we think is going on here. That is, again, with Virchow's triad on the right, that oral contraceptive use will cause a lowering of serum albumin. This is confounded by inflammation. We also saw increased inflammation in these individuals. That hypoalbuminemia can lead to a degradation of what's called the glycocalyx, which is the fuzzy coating of the inside of all of your blood vessels. And that is the right arm, if you will, of the triad with endothelial injury. We know that lower concentrations of albumin will change the fatty acid composition of the red blood cells, leading to an increased coagulability, which is a second arm of Virchow's triad. We also saw an increase in transferrin concentrations, which is another protein which is involved in clotting regulation. Again, getting at hypercoagulability. And we know during spaceflight there are hemodynamic changes, that there are differences in the way the blood flows during spaceflight. And all of these things combined to result in what we saw was a blood clot incident. And there is a concern, and there's a lot of people looking at this at NASA now, that this is something we really need to pay attention to. I moved now to one of my pictures, one of my favorite pictures. This is a bit aged now, but this is the early 1990s when we were celebrating the 500th anniversary of Columbus to find America. And those are replicas of his ships floating in the coast, off the coast of Florida, with the space shuttle Endeavour in the background. And I use this to make 2 points. One is, if we look back through the history books are rife with examples of where nutrition either made or broke exploration missions. Seafaring missions, polar expeditions, you name it. Scurvy is always the example I like to throw out. Everybody has a vague idea that scurvy was a big deal. Realize that scurvy, vitamin C deficiency, killed more sailors than all other causes of death combined. And it's important to remember that scurvy was cured by lemons and limes, but it wasn't magic. It was nutritional biochemistry, and it was the nutrients in those lemons and limes that changed the biochemistry, which changed the physiology, which allowed further exploration of Earth. And we need to make very sure that we understand the underlying biochemistry of the physiological problems that we're studying, because those are likely where the answers lie. And with that, again, I thank you for the opportunity to present here today. I leave you with an image of Sydney from space. This was taken during, I believe it was Expedition 56, just about 2-3 years ago. With that, I'll thank you again. And if there's time for questions, I'd be happy to try to answer them. Or I can just keep talking.
Sorry, didn't realize we were on mute there.
All good?
Yeah, all good now.
Thanks so much, Govinda. Thank you so much, Scott. Really so interesting and a lot to think about, not the least of which is I now need to take my advice from my nutritionist about lipids more seriously. So yeah, really good and great to have people like yourself giving time to us to consider all of these different aspects. Certainly over the last 2 days, we've been really exploring different ways for the human body to either support itself in times of need, and but also to reflect on the challenges we have about taking medicine to space. And if food is medicine, then you know, we certainly have that challenge as well, and trying to find ways to get our vitamin C in space if we're away for a very long time. I'm sure these are things that everyone is now really deeply thinking about.